Assalamualaikum dear students, this is Mudassir Mirchan. Welcome back to the playlist of cytology. In the last video of the playlist, we talked about the ribosomes, organelle which was discovered by George Palade in animal cells. Ribosomes were separately discovered in plant cells also by Robinson and the Brown. We used some terms like 70S and 80S in our video. So before we move ahead with the structure of the ribosomes, let's try to find out what this S stands for. S stands for Swedberg's unit or sedimentation coefficient. Swedberg unit is the rate at which the ribosome sediments in an ultracentrifuge. What? Yes, when we put cell contents in the test tube and rotate it in an ultracentrifuge at a very high speed, cell contents and organelles sediment at different speeds based on their mass, size, density, and viscosity, etc. The speed at which an organelle settles down or sediments in an ultracentrifuge is written in terms of Swedberg's unit. Now, when any organelle is labeled as 1s, it means it sediments at the speed of 1 into 10 raised power minus 13 seconds. Oh, that means 70s ribosome sediment at the speed of 70 into 10 raised power minus 13 seconds. That's why it's called as 70s. And 80s sediments at the speed of 80 into 10 raised power minus 13 seconds. That's why it's called as 80s ribosome. Okay, now we can move ahead with the structure of the ribosome. Well, ribosomes are not membrane-bound organelles. They are rather made up of RNA and proteins. Therefore, they are also termed as ribonuclear protein particles, RNPs. Every ribosome has a larger subunit and a smaller subunit. So, therefore, 70s ribosome has a larger subunit and a smaller subunit, and 80s ribosome has a larger subunit and a smaller subunit. Now, in case of 70s ribosome, largest subunit is designated or labeled as 50s, while a smaller subunit is labeled as 30s. Again, a very important point. Don't do the arithmetic sum of 50 plus 30 is equal to 80. No, it's not like that. It means for larger subunit of the 70s ribosome, sedimentation coefficient is 50s. And for its smaller subunit, its sedimentation coefficient is 30s separately. Now, larger subunit of the 70s ribosome, let us see what it is made up of. It is made up of 23s rRNA. That means a ribosomal RNA which is labeled as 23s, Swedberg unit. Plus it is made up of 5s rRNA. That means a ribosomal RNA which is designated as, which is labeled as 5s. 23s rRNA, 5s rRNA, and some 34 different types of proteins. This is what a larger subunit of 70s ribosome is made up of. Similarly, its smaller subunit is made up of 16s ribosomal RNA and some 21 different types of proteins. In 80s ribosome, largest subunit is 60s and the smaller subunit is 40s. Larger subunit of 80s ribosome is made up of 28s rRNA, 5.8s rRNA, and 5s rRNA, and some 45 different types of proteins. And its smaller subunit is made up of 80s rRNA and some 33 different types of proteins. Well, that was what smaller and larger subunits of the 80s and 70s ribosomes are made up of. It's important to note here that the subunits of a ribosome occur separately in the cytoplasm and join only when the proteins are to be synthesized. When a cell needs to synthesize a protein, many ribosomes line up, form a chain called as polyribosome or a polysome. In our next video, we will discuss the ultrastructure of the ribosome and the functions of the ribosome. Till then, stay blessed. Assalamu alaikum.